It's a real honor for me to introduce our next keynote speaker, Andre Nussenzweig, who is a distinguished investigator and also the chief of the Laboratory of Genetic Integrity at the National Cancer Institute in the United States. Being a distinguished investigator at the NIH is the highest honor for any investigator. We all know, I bet we all know, all the wonderful things, all the wonderful contributions that Andre has been making to the field of DNA damage repair, DNA damage signaling, and so on. But I bet you very few of you know about very interesting, background information about Andre. I'm going to tell you a little bit about him. He was born in Brazil. He actually studied as a physicist as an undergraduate at New York University. He then went to Yale University and got his PhD in physics, and even did a postdoctoral fellowship, as far as I understand, in France in physics. Somewhere along the line, he saw the light, and it was a good thing that he saw the light. He decided that he would do biology instead, so he actually did a second postdoctoral fellowship training at Sloan Kettering Cancer Institute, and then became a tenure-track staff member at the National Cancer Institute in 1996. It actually wasn't that long ago. And then has made incredible contributions to the field with numerous seminal papers in journals that we wish we'll be able to publish in. Just really distinguished career in that particular regard. He's the winner of many prestigious awards, no wonder, including the Arthur Fleming Award by the National Cancer Institute, the NIH Directors Award, and recently, the Best Global Prize in BRCA research. He's an elected member of many honorific societies, including the EMBO organization, as well as the national academies, including National Academy of Medicine and also National Academy of Sciences of the United States. Now, you're here to listen to Andre, not me, so I'm going to hand over the podium to Andre. I see that he's very eager to get up here to tell you about some of the exciting work that he has been doing. Andre.
Thanks so much. That's very kind of you, Patrick. Can everybody hear me? Yeah. It's interesting. I had a interesting experience at the airport. Identification with Patrick. Actually, you don't know about this. So when I left the customs, all I had to do was find my driver. So there was a huge line of signs. So I just looked for my name, go down the line, and I couldn't find my name. And then I see Patrick Sung. That's interesting. I continue on, and at the end of the line, I still didn't find my name. So I said, okay, I must be jet lagged. So I go back in the other direction. Still not see my name, and then notice Patrick Sung, and then to the end, not my name. What do I do? So I went to Patrick Sung and said, maybe you're here to pick me up as well as Patrick. And the fellow looks at his phone, shows me his phone, and it says, Andre Nussenzweig. It's like, okay. And he was happy to take me. So I guess you came earlier than me.
Yes, I did.
So anyways, today I would like to talk about some interesting differences that we have found when we've studied carefully replication-coupled double-strand breaks and what I'm calling canonical double-strand breaks. And this, I think, sheds light on how cells choose between repairing double-strand breaks. In other words, pathway choice. And in particular, there's a conundrum, really not well understood what the role of BRCA1 is. Steve mentioned BRCA1, but what does it actually do? And this question has been around in the field, and I thought it was interesting, if you follow Twitter, that Fina Oakes posted a question. She was discussing BRCA1 with a student project, and going through the literature, she was curious to know what people think in terms of BRCA1's function in end resection. Does it promote end resection? Does it counteract end resection? Is the role complex dependent, or does it not affect resection? So it's very confusing. And if you go through the literature, starting with 2004, BRCA1 was implicated in resection, and this was using double-strand break reporters. And this was verified later, a few years later, after irradiation, that BRCA1 is required for resection. 2010, it was found that BRCA1 overcomes 53BP1's role in blocking end resection. A little bit later, it was realized that maybe BRCA1 is not required for resection. And then a few years later, BRCA1 was found to accelerate end resection. So it's really, really not clear up until this time what is the role of BRCA1. I don't know I probably would have given the answer that it's required for resection if I had to choose. But today, I'd like to show you that I'd like to argue that the right answer is that it depends on the context, and that's why it's a little bit confusing what BRCA1 does. And this, we came about this when we compared the response to replication breaks versus canonical breaks. So let me define what a replication break is. It's basically the break requires that the replication fork runs into a lesion, let's say a nick in the DNA. That's what causes the double-strand break. And the nicks are very prominent. They are estimated to occur 1 per cell per second. So they're happening all the time because of base excision repair intermediates. Whereas the canonical breaks are much more rare. They can occur after radiation, or they've been studied extensively with double-strand break reporters. But endogenous double-strand breaks indeed are rare. Maybe they happen because of 2 nicks on opposite strands. And but this pathway has been mainly been studied, and this has led to a view about how double-strand breaks are, how the choice between double-strand breaks is made. So the canonical view of double-strand break repair is that there's 2 major pathways, as Steve mentioned, non-homologous end joint and homologous recombination. And the most rapid way to repair a break is through non-homologous end joining, where you have Ku binding, and that's followed by the end ligation by ligase 4. If this does not happen efficiently, you get the ends are resected by BRCA1, MR11, and CTIP, and this inhibits Ku binding. And this is followed by RAD51 and BRCA2 dependent homologous recombination using the sister chromatid as the template. In addition, in addition to this, it is known that 53BP1 counteracts BRCA1's role in resection at canonical double-stranded breaks. There is also a third pathway called microhomology-mediated end joining, and this happens after resection. And here, microhomologies are found at both sides, and then the ends are processed, and then the microhomology is joined through a Pol theta-mediated pathway, and this leads typically to small deletions. So this view of double-strand break repair, this canonical view, says that there's basically 3 pathways, and they compete with each other to repair the double-stranded breaks. However, the story is different at a collapsed fork, which is really the physiological trigger of homologous recombination. And I'd like to argue today that in this case, non-homologous end joining really does not play a role, does not compete. Microhomology, and not only that, but BRCA1 does not promote end resection. 53BP1 does not counter BRCA1's role, because BRCA1 does not function in end resection. And microhomology-mediated end joining is also a very, very minor pathway. It's really all about homologous recombination. There's no competition. After all, this is the most accurate way. This is the major way to repair. So there's no competition. However, in the absence of homologous recombination, then you get abundance of microhomologated end joining events. And this can be seen if you sequence tumors that are homologous recombination deficient. If you sequence a tumor that's homologous recombination deficient, the main feature, the most predictive feature of that it's homologous recombination deficient is that there are a high level of deletions with microhomology. This is a way you can even predict based on the sequencing of the tumors. So this pathway is very, very abundant, the MMEJ, only when you inactivate homologous recombination. Okay. However, there's another conundrum, another problem here, is that these deletions and rearrangements that happen in the absence of homologous recombination arise from the joining, have to arise from the joining of two ends. So as I mentioned, you expose the ends and they are resected, and then they're joined through this deletion event through Pol theta, and this requires two ends. However, the primary source of double-stranded breaks are collapse forks, and this leads to one-ended breaks. Okay? So the question is, how can a replication-borne break, which is a one-ended break, lead to an indel which requires two DNA ends, a left end and a right end? And the key assumption here is that the replication collision with a nick actually generates a one-ended double-stranded break. And so we wanted to look at this more carefully. And we really reached the conclusion that in homologous recombination deficient cells, a collapsed fork basically always generates a two-ended double-stranded break. And even in wild-type cells, you can get, under certain circumstances, you can get a two-ended double-stranded break, as I'll show you. And in addition, I will show you that BRCA1 and 53BP1 function differently at a replication-borne break versus a canonical double-stranded break. And so we set up an assay to carefully study replication-coupled double-strand breaks. And the idea is to make DNA nicks around replication origins, where the replication fork direction is very, very well known, and it's unidirectional. So you can map replication origins or initiation zones by different methods. For example, here we used EDU-seq, where we arrest cells in G1, and then we release them in the presence of a fetacolon. That doesn't inhibit initiation of fork initiation or origin firing, but inhibits the progression of replication forks. So you see these peaks when you sequence EDU, and these peaks correspond to the initiation zones. You can also look at OK-seq data that's mapped in the absence of a fetacolon, which maps replication fork directionality, and a change in replication fork direction, as shown here, corresponds to an initiation zone, and that is exactly where we found map these initiation zones with the EDU-seq. So the idea of the experiment is basically have the replication fork run into a nick in the DNA and let the fork collapse. And so we make a nick on either side of these unidirectional replication forks, and then we examine the structure of the ends. And we make a nick with a inducible Cas9 nickase. We use the D10A nickase, where the nick is on the same strand as the guide RNA, and we induce it with doxycycline. So basically, so knowing the fork direction, we can make a nick on the leading strand and ask what happens when the replication fork runs into the leading strand, or we can make a nick on the lagging strand and determine what happens when it runs into the nick on the opposite strand. And the two questions we have is, what is the end structure? What does the end structure look like for collapses, and how are these collapse forks repaired? Maybe you've would have guessed it that we will use NSeq to determine the structure of the DNA ends. And basically, with NSeq, what we do is we blunt the DNA ends, and then we ligate hairpin biotinylated adapter to the ends, and then we pull down and sequence the ends. And so you get a left end, you get negative strand reads, and the right end, you get positive strand reads. And the data basically looks like this. But if the ends are resected, when you blunt the end, your first nucleotide that you sequence is going to be at a distance from the actual break site. So you get a distance of ends. That's what a resected end looks like. If we have, and as the case here, replication fork hits the leading strand. You have a one-ended break. And then we're going to be sequencing 5' to 3'. So you're going to be sequencing the gray strand or the bottom strand, and the signal should look like this. Fork moving to the right, you get a negative strand reads. If the nick is on the lagging strand, and you also get a collapsed fork, you sequence from 5' to 3', you get the gray strand, and you basically get the same result. So whether the nicks are on the leading or lagging strand, as long as the fork is moving to the right, you get negative strand reads. If the fork is moving to the left, you get positive strand reads. That's all you have to remember. So the experimental setup is that we synchronously release cells into the nick. So we arrest cells in G1, induce our Cas9 nickase, and then release cells into S phase. And so we started with the dead Cas9, coupled to different guide RNAs, released cells into this dead Cas9 nickase complex, and we don't detect any DNA breaks with a dead Cas9 as expected. No DNA breaks. And then we released cells into Cas9 that was coupled to a leading strand guide. And you can see that the fork moving to the right, you get mainly negative strand reads. The fork moving to the left, you get mainly positive strand reads. But don't ignore that little bump over there. I'll be talking about that little bump. However, the surprising result was when we released the cells into Cas9 coupled to a lagging strand nick. And in this case, we see a symmetrical peak with bottom and top strand reads. And these are two-ended breaks rather than the one-ended breaks here. And so if you zoom in and you look in more detail what the structure of these is, you can see that these ends, whether they're two-ended or one-ended, are highly resected. So you get resection lengths of about 5 kb on either side. And you can see very clearly that the leading strand one-ended break is highly asymmetrical. It's mainly this bottom strand peak. You can measure the asymmetry. And whereas the lagging strand guides generate a very symmetrical peak. It's like exactly the same on the top and the bottom. And so the question is, why does the lagging strand collapse generate two-ended double-strand breaks? Because this was not predicted. We had our first hypothesis is this must come from a converging replication fork. So for example, like as follows. So you have a nick on the lagging strand. Replication fork runs into it, actually generates a single-ended double-stranded break, and then for some reason it's not repaired. It's not prepared, and it hangs out there. And then you have a converging replication fork coming in, and then it converges, and this second event will produce this two-ended double-stranded break, which is then resected. Okay, so let's test the model that the converging fork is responsible for the two-ended double-strand break. To test this, what we did is we tried to block the converging forks. And so we used additional guide RNA. We knew the replication fork origin, the distal origin, was on this side of these two blocking guide RNAs, and so we tried to block the converging fork. And we knew that the guides all three guides actually generated DNA breaks because we first arrested cells in G1, and then prior to NSeq, we treated the DNA with recombinant S1 nuclease, which will convert that nick into a double-stranded break. And so in G1 cells, you see these three peaks, which correspond to the three different guide RNAs. So we know that the blocking guides are working. And in fact, the second guide here is more efficient than the first guide. And so when we release cells from the G1 arrest, we actually see that the first guide gives a larger signal than the second guide, meaning that it's the first blocker, so it does a good job already, and then the second guide is a little bit lower. And the prediction was that if the converging forks contribute to the second end structure, then you would lose this top strand from forks moving to the right. You should only see this. However, what we observed is that the structure did not change at all. So basically, even if you block converging forks, you get a two-ended break. The second end does not depend on the converging replication fork. So this structure is independent of the converging fork. How about this? How about this little bump that we found was highly reproducible? So here, to study this in a more global way, we collaborated with Wei Wu, who's a former postdoc and started her lab, has been in her lab in Shanghai for 2 years, and she generated a degenerate guide RNA that would target multiple sites in the genome. And so you can see that upon DOX induction, you get these double-strand break peaks in multiple sites in the genome. And for each site, you can compare it with a replication fork direction. And you can see that some peaks are very asymmetrical. They're basically one end. And other peaks look like they're two-ended peaks. There's two flavors, one-ended peaks and two-ended peaks. And this can be seen pretty dramatically if you zoom in to one of these sites. So these are two break sites right near a unidirectional replication fork going to the left. And you can see that in the first case, you get a one-ended break, a single-ended break, and in the second place right next to it, you get a double-ended break. It's probably because these are different cells that have collapsed replication forks. So you can determine where the guide RNA is, if it's on the top strand or if it's on the bottom strand. And you know the fork direction. And we know, for example, that this guide RNA is on the top strand, and this should generate a leading strand, Nick, which generates a one-ended break. And we know that this one happens to be on the bottom strand, and it generates a two-ended break. So basically, by knowing whether the Nick is on the top strand or the bottom strand, and knowing the replication fork directionality, we can determine whether it is a leading strand break or a lagging strand break. And so we basically can put these two in different buckets. We can put all of the leading strand nicks into one bucket, lagging strand nicks into another bucket, and we can ask the question, how is the structure of the DNA end related to replication fork directionality as determined by OK-seq? And when we look at the lagging strand nicks and compare the symmetry of the ends, determined by NSeq, with a replication fork directionality, you can see that there is no relationship, really, between the replication fork directionality and the structure of the DNA ends, and the ends are highly symmetrical, are quite symmetrical, two ends. However, if you look at the leading strand nicks, you can see that the structure of the ends are highly correlated with the replication fork directionality, namely that if forks are moving to the right, you see mainly left end signal, and if left, you see mainly right end signal. And you can actually predict the structure of the DNA end based on the replication fork direction. So what I'm saying is that, for example, these two peaks that we observed comes from the majority of the forks moving to the right, like 90%, for example, and a few of the replication forks moving to the left. So these are basically a combination of one-ended breaks from fork coming independently from different directions. But still, mystery is how about this lagging strand collapse that has nothing to do with the converging forks? Where does that come from? So we were puzzled by this. We had some hypothesis, but we didn't really have any data on this. And we got a while ago, we got an email from Johannes Walter, who had heard about our observations, and he said that he had some single molecule experiments that might be related and help explain this. And this was data from his postdoc, Kyle Vrittis, and he actually had a copy of his lab meeting, which was on Zoom because it was recorded during COVID. So I watched this seminar 2 hours. It was interesting to hear other people's lab meetings. This was a 2-hour lab meeting, really fascinating lab meeting. But it really helped us solve the problem of where the two-ended breaks come from. So this is their experiment where they did the single molecule imaging. And here they're replicating a tethered DNA in xenopus extracts. And this immobilized DNA is stretched out, and they just add the xenopus extracts, but the extracts are missing the GINS component of the CMG complex, and they reconstitute with the labeled recombinant GINS, which is labeled in green, so they can track CMG, and they also track the nascent DNA with a labeled FEN1. And when they add the xenopus extracts, and with these labeled proteins, they can see that the CMG is at the leading edge of the nascent DNA. The nascent DNA is following the CMG here in green. And then they add one more component, and that is the nickase, the Cas9 nickase, together with the guide RNA. So they can track what happens with a replication. The CMG bumps into the Cas9 nickase. And so first, you'll see what happens when the nick is on the leading strand and the CMG runs into it. And you can see that in this chymograph, this is the nickase in magenta. You can see that the CMG runs into the nickase. It hangs out there for a little while, and then they both fall off. And this happens basically every single time. So it's something like this. At the leading strand collapse, they fall off, and eventually both of them are gone. And this corresponds to the one-ended double-strand break that we detect. They saw something very, very different at a lagging strand collapse. And in this case, the CMG in every case bypassed the Cas9 nickase in magenta. You see the bypass of the green past the red. The green bypasses, the green bypasses. Every single time the CMG bypasses the Cas9 nickase. So it's something like this. The CMG runs into it, it just bypasses it, and then they both fall off, and you get now two ends of the double-stranded break, as shown here. So basically, the CMG continues to unwind and bypasses the nick on the lagging strand. And in addition, they saw that there was basically a hole where the FEN1 had traversed, and this, we think, corresponds to end resection, which is exactly what we saw. So in summary, the nick on the lagging strand is bypassed by the CMG. It just continues on. And then the Pol alpha primase synthesizes past that nick, and this results in the two-ended double-stranded break, which is then resected. So in summary, the Cas9-induced nick on the leading strand generates an asymmetrical one-ended double-stranded break. The degree of asymmetry is directly proportional to replication fork directionality. And when the nick is made on the lagging strand, this generates a symmetrical two-ended double-strand break, and the two-ended break is due to CMG bypass of the lagging strand. And now I'd like to address the second question, and that is how the collapsed replication forks are repaired. And we can just do the kinetics of repair. So this is a replication origin, fork moving to the left and right. You can see that the breaks are generated early on. They are maximum at 6 hours, and then they disappear by 12 hours. So the repair in this system occurs in about 6 hours. But the ends, interestingly, are, as soon as we look, the ends are resected. The fact that the ends are basically immediately resected is consistent with the result from this publication, which showed that non-homologous end joining does not play a role in the repair of the Nick-induced fork collapse. And we think that's because the immediate resection eliminates Ku binding, and then the cells are committed to Rad51-dependent homologous recombination. So to test this, we inactivated homologous recombination components like Rad51 and examine what happens does it actually repair, and what is the structure of the ends in the absence of Rad51. And so here again in wild-type cells, the cells repair that fork within 6 hours. You get a two-ended break for the lagging strand collapse. But in the absence of Rad51, you don't get any repair, and the ends just continue to resect and resect and resect. We were surprised when we looked at the other flavor of when we had a leading strand nick, what happens. Again, in the leading strand nick in wild-type cells, they produce mainly one end, and this gets repaired. But in the absence of Rad51, it looked like not only do you not get repair, but these one-ended breaks are converted to two-ended breaks. We decided to study this in a different way by doing a chip for RPA, the single-strand binding protein, at the same one-ended collapsed fork. So here in wild-type cells, you have the nick here, and then you found that RPA binds basically on one side, on the black strand, which is the top strand, which corresponds to the 3' resected DNA end. And when we knocked down Rad51, we found that it was quite different. Now you have basically a two-ended break, and RPA is now bound to the left side and to the right side. So this one-ended break seems to be converted to a two-ended break. And we found that that's the case not only in the knockdown of Rad51, but also if you knock down BRCA2, you get a two-ended break structure, even at leading strand collapse, and the same for BRCA1. You knock down BRCA1, and you get this one-ended breaks are converted to the two-ended breaks. So now we thought maybe in this case, the second end is due to the converging replication fork. So we did the same experiment as before, where we used blocking guides to block the converging fork and see what happens in that case. And so again, in the absence of Rad51, you see these two-ended breaks, and that's without blocking the converging fork. But now when you block the converging fork, as shown here, that worked very well, you see that this second end is reduced in intensity, and this is highly reproducible. So and it becomes more asymmetric, this peak. So we think that in this case, in the absence of Rad51, for a leading strand break, the one-ended break is converted to a two-ended break through the converging replication fork. And so you can have a model as follows. You have a nick on the leading strand with the first replication fork. This generates a single-ended double-stranded break, but homologous recombination is deficient, so you don't get any strand invasion. And we think that the last Okazaki fragment, that this nick is not ligated, then the other fork runs into this and to this gap, and this generates, as a result, this second-ended double-stranded break. And we think that this is the reason why in homologous recombination deficient tumors, for example, you can get your indels from microhomology-mediated end joining because the one-ended breaks are converted to two-ended breaks through the converging replication fork. So then we were wondering, going back to that Twitter, what is the role of BRCA1 in this process? What does BRCA1 do? So we thought that BRCA1 basically opposed the role of 53BP1 at the level of resection, promoting resection, and inhibiting 53BP1's block to resection. That's what happens at a canonical double-stranded break, but let's see what happens at a collapsed fork. So in wild-type cells, you see again that the breaks are repaired and they're highly resected. The surprising result was in the BRCA1 deficient cells, if you look at the end resection, they're just as much or even more resected than wild-type cells if you quantify this, and they're not repaired. So end resection is not affected at all in the BRCA1 deficient cells. And we confirmed this is one site, and we also wanted to look throughout the genome and at if you were in the transcription or non-transcribed region. So we generated another degenerate guide RNA targeting ALU elements that should target hundreds of thousands of sites in the genome with one guide RNA. And then we induced this nickase, and this is the structure of the ends we found. There are breaks all over the place in wild-type cells and BRCA1 deficient cells. And if you zoom in here at any of the breaks, you can see that the ends are highly resected in the absence of BRCA1, even more so than wild-type cells if you quantify it. The maximum resection length is even greater in BRCA1 deficient cells. Again, for a collapsed fork, BRCA1 seems to be dispensable for end resection. That's with the nickase Cas9. And so we decided to use the same guide RNA, but with a wild-type Cas9 to see what happens at a canonical break. And when you do that when you generate a canonical break, you can see that the end resection is much reduced in the BRCA1 deficient cells. So here in this case, BRCA1 seems to be required for end resection when you have a canonical break, different from a collapsed replication fork. So what is the role of 53BP1 and BRCA1 if BRCA1 is not required for resection, but yet it's required for repair? See, the ends are not repaired in the absence of BRCA1, and then we inactivate 53BP1, and now the ends are repaired. But this is independent of end resection. So we have to think about the opposing roles of BRCA1 and 53 a little bit differently. So they can't be acting over here, but we know that BRCA1 also promotes, through its interaction with PALB2 and BRCA2, the loading of Rad51 and Rad51 filament formation. So we thought, okay, maybe actually BRCA1 is opposing 53BP1's block, not to resection, but to the loading of Rad51. And so we thought we have to examine Rad51. So we did a chip for Rad51 at the collapsed forks, and you can see that you can get very, very abundant Rad51 signal at a collapsed fork. This is reduced in the absence of BRCA1, but when we inactivate 53BP1, the signal reappears. So it seems that 53BP1 is acting by blocking the loading of Rad51 in the BRCA1 deficient cells. What could be the mechanism by which 53BP1 blocks Rad51 loading in BRCA1 knockout cells? How does this work? Well, we think that it's mainly related to a competition of binding to H2A lysine 15 ubiquitination. Data from Ross Chapman's group has shown that BRCA1 binds to chromatin through the interaction with BARD1, which directly interacts with lysine 15 ubiquitinated on H2A. And this mark is mediated by RNF168. if BRCA1 binds to this mark, it inhibits 53BP1 binding to the same mark, H2A lysine 15 ubiquitination. And RNF168 not only ubiquitinates H2A, but it also interacts with PALB2 and BRCA2, and that's been shown by Heiko van Adicum. And so in BRCA1 deficient cells, now 53BP1 binds to lysine 15 ubiquitination, but that inhibits RNF168 binding to its own mark. And then in BRCA1 53BP1 deficient cells, now RNF168 can be recruited and generate this alternative mode of RAD51 loading. Can I test this experimentally? Well, we take BRCA1 53-1 deficient cells and then look at RAD51 loading, which I showed you is quite abundant, and just inactivate RNF168. And this is what we observed. In BRCA1 53-1 deficient cells, when you inactivate RNF168, you see a reduced level of RAD51 binding. You can do this experiment really simply by just treating the cells with camptothecin as well. In RAD51 forms foci in response to camptothecin. In BRCA1 deficient cells, those foci are severely reduced. They're rescued in the absence of 53BP1, but when you inactivate this ubiquitin ligase, RNF168, they basically go back down to 0. So in conclusion, BRCA1 is essential for resection at canonical double-strand breaks, but is dispensable for resection at collapsed replication forks. Here we have evidence that CTIP is required. The rare pair of collapsed fork requires homologous recombination proteins, and in their absence, one-ended breaks are converted to two-ended double-strand breaks by adjacent replication forks, and this leads to the genomic scars that are observed in homologous recombination deficient cancers. The physiological role of BRCA1 is really to promote RAD51 loading on resected single-stranded DNA, we think, and 53B1 doesn't block resection at collapsed forks, but inhibits RAD51 loading in BRCA1 deficient cells through a mechanism involving RNF168. So what is the fundamental difference between a canonical double-strand break and a replication-coupled double-strand break? We think that the bottom line is that when you have a canonical double-strand break, everybody has to be recruited to the break site. The resection machinery, BRCA1, 53BP1, and that leads to a competition between non-homologous end joining and homologous recombination, and BRCA1 opposes 53BP1's role in blocking resection at canonical breaks, but in addition, it opposes the role of 53BP1 in blocking RAD51 loading. We think that the difference really is that the resection machinery, we think, travels with the unperturbed replication fork. It is ready to promote resection and homologous recombination in case there is a nick in the DNA, for example. In this case, the resection is independent of BRCA1 because the resection is carried out by the resection machinery that's traveling with the fork. And BRCA1's, really the role is to oppose 53BP1's block to the loading of RAD51. And I just want to acknowledge the people that did the experiments here. This work was led by Rafael Pavani and Vinu Tripathi in the lab, and they got help from many others in the lab. And we collaborated, as I mentioned, with Wei Wu and her group in Shanghai. And the single molecule imaging work was done by Kyle Vrittas in Johannes Walter's group. So thank you very much, and happy to take any questions.
But there's quite a lot of literature on toxic end joining, and the model there is thought to be that Ku binds one of those single-ended collapse breaks and channels that into some kind of aberrant translocation or recombination event at a similar site across the genome. Your data, your feeling is that wouldn't fit. That wouldn't fit with your model because you think that HR is the only way that those breaks are processed. So how do you think toxic NAGJ is happening?
Well, the context, I guess, that I've seen toxic NAGJ is, for example, in ATM deficient cells treated with camptothecin, if I'm not wrong. And there you get toxic NAGJ. And the idea originated from Steve Jackson was that ATM deficiency blocks a little bit of resection, and so you can get toxic end joining. It might happen, but in wild-type cells, but it's very rare because, probably these nicks and collapsed forks could happen very frequently, and they need to be repaired accurately. So I would just think that toxic end joining is just...
It might be, I don't know if you've looked, it might be worth seeing if you can chip COO at those leading strand single-ended events.
Yeah.
And whether or not they ever get in, if it's possible to do, whether or not they ever get involved in weird translocations, because it does happen in wild-type cells. So, for example, in DT40, Alfred in the center showed a long time ago that the sensitivity of part 1 knockouts to camptothecin, for example, can be partially rescued by deleting COO. And in Steve's work, it was ATM dependent, and he rescued it by deleting LIG4. So it does imply that if some of these breaks, even in wild-type cells, COO gets a hold.
It's possible. And we haven't done the chip. We should do the chip. But the one thing is that the ends, as soon as we look, they're resected. So we hypothesize, but we haven't done the experiment, that the resection would inhibit COO binding. And also there's data, as I mentioned, from Maria Jason, which if you normally have a canonical break, if you inhibit NHEJ, you get increased HR. At a collapsed fork, you inhibit NHEJ, doesn't have any effect on HR. So they're different, but we haven't actually done.
It might be a very rare event, but nonetheless.
Yeah, I think it's very rare.
From a toxicity or a genome stability endpoint, it might be significant.
So, if I may, great talk, wonderful, beautiful work. Do you know which nuclease or nucleases are resecting the one-ended break?
We only know that CTIP is involved, but we haven't actually tested.
CTIP upregulates 601 as well as BOM and Werner.
Yeah.
So it could be all three pathways?
Could be, but we have not yet figured that out.
Just very quickly, have you thought about whether the E3 ligase activity of BRCA1.1 may be required?
The ligase activity.
It could be degrading things that inhibits RAFD1 loading, in addition to competing for the epigenetic mark.
Yeah. We haven't tested that.
Yeah,
but that's certainly
fascinating. Okay, great.
So
lastly, and shamelessly, I'm going to promote myself a little bit. I'm going to present data tomorrow morning about a role of BRCA1.1 in upregulating XO1, BLM, and Werner at a canonical break.
Okay. So we'll learn more about end resection at canonical break tomorrow.
Andre, so RNA-168 is required for the recruitment of 53BP1, right?
Yeah.
With the H2A. So in your model, you have 53BP1 inhibiting RNA-168 also in the way you...
Well, it's inhibiting the binding of RNF-168 to its own mark.
Right, but RNA-168 gets recruited, ubiquitinates histone H2A, and then gets removed by 53BP1? That's what you're proposing.
Yeah, actually, we can actually track RNF-168 foci plus or minus 53BP1, and at a collapsed fork, you don't see RNF-168 recruitment, but you inactivate 53BP1, you can see RNF-168 foci. So we think there's a competition between 53BP1, in fact, BRCA1 and through BARD1 and RNF-168.
So RNF-168 comes and very quickly gets removed by 53BP1.
Correct. You treat with camptothecin, you know that you get H2A ubiquitination, but you don't get RNF-168 foci. You only see it in the absence of 53BP1.
So it's just the stability of RNA-168 at the foci is regulated by 53BP1. That's what you're proposing.
Yep.
Okay, Andre, it's great work. Actually, our work published in 2022 is quite consistent what we are finding here. So in that work, we use Cas9 DNA and 60A mutant to induce the leak.
Yeah.
And then to look at function of BRCA1.
Yeah.
And then we did find the 2-ended double-strand break doesn't induce mutational signature in BRCA1 mutant cells. But leak-induced break can induce BRCA1-linked mutational signature, including indels and translocations and tandem replications.
Exactly.
So a question, and also this work helped me to answer some question I had in that work also. But in this work, in the model you proposed, you use the Cas9 and then always released with the target DNA with guide RNA. So because we know the Cas9 actually can cut the DNA, and then some of them will bind there tightly for a long time. But for some of them, they will release quickly. So what if your nick encounter different leak? One is a pure leak, another is a Cas9 bound with a leak. What happened? Are they the same?
That's a great question. And so we use the other Cas9 that nicks on the opposite strand and releases the 3' end. And here, we actually did not observe collapsed replication forks. Why? And that comes from the work from Petr Čeka, who's shown that Cas9 is actually removed from through the action of HLTF, the translocase. And so what we observed when we used this other flavor is basically very little or no collapsed replication forks. But when we inactivated HLTF and Cas9 remained bound, and then we detect a signal. So it does depend on the stability. And I should also mention that work from Ralph Scully, that he posted in bioRxiv, he used a different system, the flip nickase, which mimics TOP1cc, and he concluded as well, and with this flip nickase, that you get 2-ended double-strand breaks, and that these are repaired by a mechanism independent of BRCA1-mediated resection. So not only at Cas9 break, but the flip nickase, you get very similar results.
Yeah, we had a similar, very similar finding, but our paper kept delayed for a long time. I don't know what happened, but anyway.
Bottom line.
No, it's not. But so another question I wonder, because you use siRNA RF168 to demonstrate it's so basically you kick out of the RF168, you don't have a PowerB2 BRCA2 to recruit RAD51. Is there any other specific way rather than siRNA RF168? So basically you just block interaction between RF168 between RF168 and the PowerB2 rather than use siRNA. Is there any other specific way to do that?
To block the interaction between PALB2 and RF168.
Yes, rather than use siRNA.
There should be a way to do that. We haven't done it. We just did siRNA and also knockout of RNF168.
Because RNF168 is have a quite important function in other way, right? I'm just curious. I also have the same problem with the siRNA RNF168. So that's why I always wonder, is there another specific way to do it?
Well, we knocked out RNF168 as well, but we didn't study the specific interaction with RNF168 and PALB2.
Okay.
For example.
Okay. Yeah, that's all.
Yeah.
Thanks.
Yeah.
Hello. Do you check the DOSE DSP negation or translocation to the other DSP? Just translocation to the other DSP. Do you check is the negation mechanism rely on the NIH?
Did we observe translocations at
translocation or negation other DSP?
With other double-strand breaks, we have not done that. When we make multiple double-strand breaks at collapse fork, whether they join together.
Do you check the negation mechanism? Or
well, we haven't done that, unfortunately.
Why do you knock out the negative fork to check the DSP repair efficiency in your system. Next 4, the canonical NHJ factor.
Have we tested the role of ligase 4 in the repair? Is that the question?
So your PPT, you show no compression on HJ. I don't know why. So I want to know you look out if you look out the next 4 to check the
ligase
repair
4.
The next 4.
No, we haven't done CHIP for NHEJ. We haven't knocked out NHEG components, but clearly, in the absence of HR, the ends persist. The ends get highly resected, and they don't get resolved. So basically, HR is the main pathway. There could be minor contribution, but the ends just persist. They're not repaired in the absence of HR.
In the BRCA1 lookout cell, is the repair mechanism rely on the NHEJ?
We think that in the BRCA1 deficient cells, when you get the two-ended breaks, the repair mechanism probably involves microhomology-mediated end joining, and that's how we get deletions consistent with the previous person asking the question with where you get indels.
Thank you.
Andre, beautiful talk. I was wondering why resection happens so quickly. So is it possible that resection occurred at the nick before it becomes a break?
Yeah.
But you only see it with NSeq after it is converted to a real break.
You know, all I can say is that if you generate a nick in G1, for sure it's not resected. Whether so it definitely requires S phase, and whether the resection happens before the fork runs into it or after, I guess hypothesize that it's after because our model is that the resection machinery travels with the fork, so it just has to reach the nick beforehand. Otherwise, the resection machinery would have to see the nick and get recruited to it. So it's just simpler to think that it travels with it and then it collapses and then it's resected.
Thank you.
Yeah. Okay. Well, I guess everyone can go to sleep now. Yeah. Go to the bar. Thank you.